Chapter 9, DNA-Based Information Technologies, Part 3. In this lecture, we will discuss about DNA-based methods to understand protein function. Protein functions can be described on three levels. Phenotypic function describes the effect of a protein on the entire organism. For example, loss of a protein from an organism may lead to slower growth of that organism or an altered development pattern or even death of that organism. Cellular function. Cellular function is a description of the network of interactions a protein engages at the cellular level. For example, a protein may interact with other proteins or DNA molecules or may be involved in a metabolic process. This constitutes the cellular function of a protein. Finally, molecular function, which refers to the precise biochemical activity of a protein, including details such as the reactions of an enzyme catalyzed or the ligands a receptor binds. Challenges in understanding the functions of thousands of uncharacterized or poorly characterized proteins found in a typical cell has given rise to a wide variety of DNA-based methods. And these DNA-based methods are used to understand the function of a protein. Some of the common methods used are construction of DNA libraries from coding regions. And this method is useful in converting an RNA to a DNA. And this is very similar to the RT-PCR experiment that we discussed before. And in this case, the DNA of a live cell can be readily monitored. Using immunofluorescence to reveal protein location in vivo, one can use antibodies that are fluorescently tagged. And these antibodies that are specific to a protein of one's interest can be utilized to reveal the location of that specific protein in a cell. We will look at techniques to study protein-protein interactions. One classic example is a yeast hybrid system. DNA microarray analysis can be used to understand RNA expression patterns. And finally, editing gene functions is possible using a newly developed technique called CRISPR. And CRISPR is a technique that received Nobel Prize very recently. The first method is construction of DNA libraries from coding regions. Efforts to define gene or protein function often make use of more specialized libraries. An example is a library that includes only those sequences of DNA that are expressed. This means that DNA that is transcribed into RNA in a given organism, or even just in certain cells or tissues. Such a library lacks any genomic DNA that is not transcribed. So in this case, to make specialized DNA libraries, one typically extracts mRNA from an organism or specific cells, in this case eukaryotic cells, and then prepare complementary DNA. How do one go about in preparing a complementary DNA? And here are the steps. All mRNA molecules have what is called as a polyadenine tail. And the function of this polyadenine tail is that it helps in purification of mRNA, and it serves as a universal template. That's very important. A DNA strand can be synthesized from an RNA using that mRNA as a template. And this is catalyzed by reverse transcriptase. Again, this technique is very similar to the reverse transcriptase catalyzed PCR. In fact, the very technique RT-PCR is used in this case. The end result is a hybrid where the DNA strand is complementary to mRNA. And the hybrid can be then converted to duplex DNA, known as complementary DNA. So, how can we simplify these steps? Let me illustrate this using a diagram. Here is a detailed explanation of the steps that I just outlined. Shown here is an mRNA, and it's just a graphical representation. And this mRNA could be one that is isolated from a eukaryotic cell, a growing live cell. Now, this mRNA is transcribed from the DNA. And what our goal is to get the sequence of the DNA that this mRNA is transcribed from. So this is essentially a reverse engineering process, and hence the name reverse transcriptase. Now, this mRNA can be used as a template. And for this purpose, a primer, a poly-T primer, is used. The reason is because mRNA has a poly-A tail. So this primer would anneal to the poly-A tail once the temperature is decreased or at a specific temperature for annealing of this primer to this mRNA. Following this, reverse transcriptase is added and specific nucleotides are added so that this reverse transcriptase can convert this RNA to a DNA. And in this case, deoxyribonucleotides are added so that the strand that the new strand that reverse transcriptase is making from the mRNA is a DNA. Once the new hybrid strand is formed, this is a hybrid because this is a double-stranded, and double-stranded, one of the strand, the 5' to 3' strand is an RNA, and the 3' to 5' strand is a DNA. And why is this hybrid? Because reverse transcriptase just made the bottom strand from the top strand using poly-T primer and deoxyribonucleotides. Once the hybrid strand is made, the mRNA portion of this hybrid, which is the green portion, is degraded with alkali. And this results in just the DNA strand, which is from 3' to 5'. Now, to prime synthesis of a second strand, an oligonucleotide primer of non-sequence is often used, and it is then subjected to PCR. And when subjected to PCR, as we have seen before, a DNA polymerase is usually used and dNTPs. And once we use this, we get a duplex DNA, and in this case, a double-stranded DNA. And this double-stranded DNA is usually the DNA structure or the sequence of DNA that the original mRNA has been transcribed from. So this reverse engineering works in this fashion. And this is how cDNA or complementary DNA are constructed. Fusion proteins and immunofluorescence can reveal protein location in cells. Often, an important clue to the function of a protein comes from determining its location within the cell. For example, a protein found exclusively in the nucleus could be involved in processes that are unique to that organelle, such as transcription, replication, or chromatin condensation. To answer this question, there are two different methods that can be used. And in both cases, fluorescence is a common theme. In the first method, green fluorescent protein can be used to tag with a protein of interest. And in this case, recombinant DNA technology is used to attach GFP to the protein of interest, and it is visualized via a fluorescent microscope. In the second technique, immunofluorescence is used. Here, the antibody that is specific to a protein of interest is tagged with a fluorescent molecule. And in this case, tag protein with primary antibody and detect with secondary antibody containing the fluorescent tag. Proteins can also be fused to a short epitope, and the primary antibody detecting the epitope can be fluorescently labeled. The first technique, that is the classical method of immunofluorescence. The second method is slightly advanced, and we'll talk about both these methods in the upcoming slides. Green fluorescent protein is derived from a jellyfish, Aquaria victoria. The protein has a beta barrel structure, and the fluorophore is in the center of the barrel. The fluorophore in GFP is derived from a sequence of 3 amino acids, tyrosine, serine, and glycine, as shown here. The fluorophore achieves its mature form through an internal rearrangement that is coupled to a multi-step oxidation reaction. An abbreviated mechanism is shown here. An example of GFP-tagged protein localization is shown here. What is shown here is a nematode worm. And in this nematode worm, a researcher is observing the location of a protein GLR1 or glutamate receptor. GLR1 is tagged to a GFP, and the green colored dots are GLR1 GFP fusion proteins, and it shows the location of this protein. The magenta colored droplets are autofluorescing fat molecules. On the right, the membranes of E. coli are stained with a red fluorescent dye, as seen here. The cells are expressing a protein that binds to a resident plasmid that is fused to GFP. The green spots indicate the location of these plasmids. So this is a typical example of how GFP can be used to visualize proteins in cells. In many cases, visualization of a GFP fusion protein in a living cell is not possible or practical or even desirable. The reason is that the GFP fusion protein may be inactive or may not be expressed at sufficient levels to allow visualization. In this case, immunofluorescence is an alternative approach for visualizing the endogenous proteins. This approach requires cell fixation and thus death of a cell. Hence, this specific methodology cannot be performed on live cells. The protein of interest is bound to a primary antibody, and a secondary antibody is added. This second antibody, with one or more attachment of fluorescent dyes, binds to the first or the primary antibody. Multiple secondary antibodies can bind the primary antibody if one requires to amplify the signal. If the protein of interest is in the interior of the cell, cell fixation is absolutely necessary. Now, what are the results of such an experiment? The end result is an image in which bright spots can be seen, which indicate the location of the protein or proteins of interest inside the cell. The images that are shown here, there are 4 of them that are shown here, is of a nucleus from a human fibroblast. And they are successively stained with antibodies and fluorescent labels for DNA polymerase E, for pCNA, which is a polymerase accessory protein, BRDU or bromodioxyuridine, which is a nucleotide analog that binds to DNA specifically. And this specific reagent BRDU is added as a brief pulse, identifies region undergoing active DNA replication. So wherever you see BRDU stained images, you see active DNA replication. And finally, a merge of all three of them. Now, the pattern of staining show that DNA polymerase and pCNA co-localize to regions of active DNA synthesis. And that is visible in this specific box here. And that's where the merge of the green, blue, and the red can be seen. Highly specialized cDNA or complementary DNA libraries can be made by cloning complementary DNAs or complementary DNA fragments into a plasmid that fuses each cDNA sequence with a sequence for a marker called a reporter gene. The fused gene, as shown here, is often called a reporter construct. Cloning of a cDNA next to the GFP gene creates a reporter construct as shown here. Transcription proceeds to the gene of interest, that is the inserted cDNA, and the reporter gene, that is GFP, and the mRNA transcript, the whole of this, is expressed as a fusion protein. The GFP part of the protein is visible with the fluorescent microscope if one were to look through it. Now, as you can see, wherever you see a green dot represents the GFP protein that is fused to the protein of interest, that is expressed as a cDNA in this case. Although only one example is shown here, thousands of genes can be fused to GFP in similar constructs and stored in libraries in which each cell or organism in the library expresses a different protein fused to GFP. If the fused protein is properly expressed, its localization in the cell or organism can be assessed. However, one of the limitations of this approach is that sometimes GFP fusion does not always work. And that is one of the main reasons why immunofluorescence is used. Another key to defining the function of a particular protein is to determine what other cellular components it binds to. In the case of protein-protein interactions, the association of a protein of unknown function with one whose function is known can provide a compelling implication of a functional relationship. We will discuss three different methods to identify protein-protein interactions. Protein-protein interactions can be identified by purifying protein complexes. And protein complexes can be purified by fusing the gene encoding a protein under study with the gene for an epitope tag. Now, the epitope tag is something that is fused to the protein of interest. The cDNA, that is the yellow box shown here, is the gene for the protein of interest, and it is attached to an epitope tag. Now, researchers can precipitate the protein product of the fusion gene by complexing it with the antibody that binds to the epitope tag. So, gentle isolation of epitope tag protein can be performed, and this can isolate stably interacting proteins. This process is termed as immunoprecipitation. All proteins isolated can be separated and identified. The immunoprecipitation process has many variations. Let us discuss the classical method. In the classical method, the gene of interest, shown in the yellow box, is cloned next to a gene for an epitope tag. And the plasmid is then expressed in a cell which expresses the protein of interest that is tagged to the epitope tag. Next, a cell extract is made and then precipitated using the antibody for the epitope tag. And the precipitate, which is called as the immunoprecipitate, is then separated on an SDS-PAGE gel. And the proteins separated are then immunoblotted onto a membrane, and proteins are then analyzed using antibodies for the protein of interest. And when analyzing using antibody for the protein of interest, other proteins that are bound to this protein of interest can be visualized in the immunoprecipitate sample. This suggests that the protein of interest is interacting with unknown proteins that are shown here. The advantage of this method is that proteins that interact with the protein of interest can be identified, or to say, can be detected using this method. The identity of these proteins are still unknown. One of the limitations of this process is that false positives can be observed. And in that case, another protein interaction method called as Tandem-Affinity Purification can be tried. And this method is called as a tap-tag method. The Tandem-Affinity Purification tag, or the tap-tag method, minimizes false positives. Two consecutive tags are fused to a target protein. The target protein is shown as orange circles, and the two different tags are attached to it, and these are called as a tap tag. One of the fusion tag is protein A, a protein that is found at the surface of the bacterium Staphylococcus aureus that binds tightly to mammalian immunoglobulin G. The second tag that is shown here is often a calmodulin binding peptide. Now, once the protein of interest along with these tags are expressed in a cell, the crude cell lysate is then passed through a column for which the matrix is IgG antibodies. Now, these IgG antibodies that makes the matrix of the column binds protein A. Most of the unbound proteins pass through the column. The proteins that bind to the IgG beads are those that have protein A attached to them. The next step is using a TEV protease that cleaves between protein A and calmodulin binding peptide. And once this is cleaved, the protein that is attached to the calmodulin binding peptide is then released from the column with IgG beads. Once it is released, it is then passed over a calmodulin affinity column. The calmodulin beads that is bound to the calmodulin affinity column selectively binds the calmodulin binding peptide that is attached to the protein of interest or the target protein. And in this case, loosely bound proteins elute. The proteins that are tightly bound to the target proteins stay with the target protein, and once the elution is complete, the final protein complex will have the target protein along with proteins that bind to the target protein. One of the advantages of this method is that it eliminates false positives. It allows two purification steps, eliminating loosely associated proteins and minimizing non-specific binding. A sophisticated genetic approach to defining protein-protein interactions is based on the properties of Gal4P protein, which activates the transcription of Gal genes, or the genes that encode for galactose metabolism. Gal4P protein in yeast has two domains that are shown here in blue arrows. The first domain is a DNA binding domain for Gal-P. The second domain is a Gal4P activation domain. The DNA binding domain binds a specific DNA sequence, and the Gal4P activation domain activates RNA polymerase to synthesize mRNA from an adjacent gene. The two domains of Gal4P are stable when separated, but activation of RNA polymerase requires interaction with the activation domain, which in turn requires positioning by the DNA binding domain, as shown here. Hence, the domains must be brought together to function correctly. In yeast 2 hybrid analysis, the protein coding regions of the gene to be analyzed are fused to the yeast gene from either the DNA binding domain or the activation domain. And the resulting genes express a series of fusion proteins. If a protein fused to the DNA binding domain, like protein X, interacts with a protein that is fused to the activation domain, or protein Y, then transcription is activated, which means that RNA polymerase is active. The reporter gene transcribed by this activation is generally one that yields a protein required for growth or an enzyme that catalyzes a reaction with a colored product. So, the interaction between protein X and protein Y is usually monitored by the action of RNA polymerase. That's what this means. Next, to perform an experiment, one of the yeast strain is grown with Gal4P DNA binding domain fusions and another with Gal4P activation domain fusions. And then these two yeast strains are mated to produce diploid cells, and they're plated on medium requiring interaction of the binding and activation domains for cell survival. All surviving colonies have interaction fusion proteins, and sequencing of the fusion proteins in the survivors reveals which proteins are interacting. And this is how yeast 2 hybrid analysis is used to identify protein-protein interaction. Major refinements of the technology underlying DNA libraries, PCR, and hybridization have come together in the development of DNA microarrays. DNA microarrays allows the rapid and simultaneous screening of many thousands of genes. DNA microarray shows differences in gene expression. Microarray chips contain fragments from genes in the group to be analyzed. The full genome of bacteria or yeast or protein encoding families from larger genomes can be used. mRNA or cDNA from different samples are differentially tagged. Analysis on the same chip shows major differences. A microarray can provide a snapshot of all the genes in an organism, informing the researcher about the genes that are expressed at a given stage in the organism's development or under a particular set of environmental conditions. For example, mRNAs from organisms at two different stages of development can be isolated, and this mRNA can then be converted to cDNAs using reverse transcriptase. Now, in this case, reverse transcriptase uses fluorescently labeled deoxyribonucleotides when two cDNA samples can be made so that one fluoresces red and another fluoresces green. The cDNA from the two samples can be mixed and used to probe the microarray. Now, in this case, each cDNA anneals to one spot corresponding to the gene encoding the mRNA that gave rise to the cDNA. The results are shown here. The spots that fluoresce green represent the genes that are under one development stage. And those that fluoresce red represents genes that are expressed at higher levels at another development stage. So green represents one and red represents another. But they are representation of higher gene expression at two different stages. Now, if a gene produces yellow, this means that they are equally abundant at both stages of development. By using a mixture of the two samples to measure the relative rather than the absolute sequence abundance, the method corrects for inconsistencies among spots in the microarray. The spots that fluoresce provide a snapshot of all the genes that are being expressed in the cells at the moment they were harvested. For a gene of unknown function, the time and circumstance of its expression can provide important clues about its role in the cell. And this is how microarray can be used to understand gene expression at various developmental stages. DNA microarrays have many applications. DNA microarray allows simultaneous screening of many thousands of genes. This is called as high-throughput screening. What are the applications? First, genome-wide genotyping. This means which genes are present in this individual. To understand that, DNA microarray can be used. Tissue-specific gene expression. Which genes are used to make proteins? Mutational analysis. Which genes have been mutated? All these are applications of DNA microarray. One of the most informative paths to understanding the function of a gene is to change or mutate the gene or to delete it. And editing the gene function can be performed by a technique called CRISPR. What is CRISPR? The CRISPR-Cas system was originally discovered in bacteria. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. And as the name suggests, these consist of a series of regularly spaced short repeats in the bacterial genome. And as you can see, this is the structure of a CRISPR-Cas9 complex. A Cas or CRISPR-associated protein is a nuclease, which means that it can cut nucleotides. The CRISPR sequences and Cas9 proteins are components of a immune system in bacteria that allowed the bacteria to evolve so that it can survive infection from bacteriophages. Bacteriophages are small virus-like particles that can invade a bacterial cell. So when a bacteriophage invades the bacteria, it spits its DNA into the bacterial cell. Now, the viral sequences are in effect spacer sequences, and it gets incorporated in the bacterial chromosome. That is called as a CRISPR locus, as shown here. Now, what happens is that this CRISPR locus has this sequence for the invading DNA that is shown in red. When the same bacteriophage attacks again, a bacterium with the corresponding CRISPR-Cas system recognizes this spacer. And sometimes these spacers are split into small RNA-like molecules called as guide RNA. And these guide RNA have adjacent repeat sequences. A guide RNA sometimes forms a complex with one or more Cas proteins or with other RNA molecules called as trans-activating CRISPR RNA. The mechanism by which bacteria recognizes invading pathogens is a little complex. What this specific diagram illustrates is that the red sequence in these repeats, or the guide RNA, gets recognized by the Cas9 complex, which originally is inactive, but when bound to this guide RNA becomes active. And using this guide RNA, it reads the sequence of the invading DNA, and the nuclease cuts the invading DNA by recognizing it and thereby inactivating it. So once the invading DNA is recognized, it automatically gets inactivated by the CRISPR-Cas9 system in bacteria. So this is the original CRISPR-Cas9 system that is now used as a tool to edit genome. But how is this tool used to edit genome? Let us discuss that. The current CRISPR-Cas9 system is adapted from the bacterium Streptococcus pyogenes. This system requires only a single Cas protein, Cas9 to be specific, to cleave DNA because it is the nuclease. Work in many laboratories, particularly those of Jennifer Doudna, who received the recent Nobel Prize, and Emmanuel Charpentier, has produced a streamlined CRISPR-Cas9 system composed of just one protein, that is Cas9, and one associated RNA, consisting of a guide RNA and tracer RNA fused into what is called as a single guide RNA, that is shown here. Now, Cas9 has two separate nuclease domains, which means that it can cut double-strand DNA. Now, this specific activity of Cas9 has been utilized by researchers where they have mutated Cas9 to have only one nuclease domain active. And I'll explain it to you in a bit as to why this is important. Now, if you consider this particular figure here, what this suggests is that the gene encoding the Cas9 protein and the single guide RNA are introduced into a cell where a target genomic change is planned. The single guide RNA has a region complementary to chosen genomic target sequence that is shown in purple. A complex, once this CRISPR-Cas9 plasmid and a recombinant fragment, which I'll explain it to you in a bit, are introduced into the cell by electroporation, the plasmid directs synthesis of the CRISPR-Cas9 complex. And so the protein is expressed. Now, a complex consisting of CRISPR single guide RNA and the Cas9 protein forms within the cell and binds to the chosen target site in the DNA. Now, two different things can happen. Once it binds the target, if the nuclease Cas9 has both nuclease domain active, it creates a double-strand break based on the single guide RNA. And the double-strand break is usually repaired by the non-homologous end joining in eukaryotes, which generally deletes or alters the nucleotides at the site where joining occurs. Alternatively, if the Cas9 protein has a mutation where one of the nuclease domain is inactivated, it can only make single strand break. And once a single strand break is made, the recombinant fragment comes into picture. As you can see, the red portion of this recombinant fragment has nucleotides that is complementary to the nucleotide that is shown here in the purple. Now, this can be utilized in many ways. If one decides to change nucleotides so that they can insert mutation, that can be done. And when the recombinant fragment is present, it sort of is used by the non-homologous repair system in eukaryotes, and it pastes this specific fragment between the cut sites. And once it's done, recombination leads to the formation of a repaired double-stranded DNA. Now, this system has immense application, and many applications are currently being developed. Potential therapeutic uses are still many years away, but developments are pointing the way to future treatment for genetic diseases such as HIV or other diseases that are genetic and are difficult to treat by conventional medicine.